Hi guys, so today you should be doing this Wednesday, April 8th. This is 3.6 Meiosis, and the color is yellow, so in your virtual notebook, this is where you're going to be writing on this yellow part of the document. Alright, so we are finishing the other part of the cell cycle. So we talked about mitosis yesterday, and today is meiosis. They sound very similar except for that I. Okay, so here are a bunch of types of cells in the body. You can see there's stem cells, bone cells, blood cells, muscle cells. They all look very different, and they all have very different purposes. So even though all of these cells have different purposes, they have identical DNA. They are somatic cells. Okay, so these cells all go through mitosis to make more of themselves as needed. So most important things, they are somatic cells, which means cells of the body. They have identical DNA, even though they're pretty different in terms of their functions. And they all make more of themselves through mitosis. Mitosis with a T. However, there are a few cells in your body that have a different agenda, and these cells are meant to create an entire new person to pass on your genes to a future generation. So here's the sperm, here's the egg. These are not somatic cells. These are not regular body cells like all of these. They are reproductive cells, and specifically sperm and egg are gametes. Okay, so are offspring genetically identical to their parents? What do you think? No, they are genetically unique. So in these organisms, humans, dogs, cats, as well as pretty much all the animals that you can think of, they're not going to be genetically identical to their parents. And we're going to talk next week about sexual versus asexual reproduction, and some animals and organisms are actually genetically identical to their parents, but not any of these. Okay, so here is that cycle again. So remember we talked about the diploid zygote, where we all started. Then mitosis and development, you grow. Okay, now you're an adult and you hit puberty, and it is time to move into the next stage. So meiosis is the process that is making gametes to make new organisms. So you already know, mitosis is cell division for body or somatic cells, and meiosis is cell division for reproductive cells. Okay, so here's where our Cornell notes are going to start. Key point number 1, meiosis is the production of gametes. So that's a new word, say gamete. Gametes are haploid reproductive cells, sperm and eggs. So haploid, remember that means half, so that's just one copy of each chromosome. Reproductive, reproduction, reproduce, making the next generation. There is a picture, a microscopic picture of sperm. There is a microscopic picture of sperm fertilizing an egg, so you can see the size difference. And then this is the same thing, but I don't think this is a microscope. This is a drawing or painting. Key point number 1, meiosis is the production of gametes. So testes, those are the male reproductive organs where meiosis happens and sperm is made. Here are the testes. Ovaries, that is the female reproductive organ where meiosis happens, and eggs are made and matured. So notice the difference between made and matured, because even as an embryo, eggs are made, but then they are matured, whereas for male, sperm is made really consistently throughout the whole entire life. Almost. Okay, so here's male and female reproductive systems. You can see that they're kind of similar structurally. And here's that picture again, because now we are comparing mitosis and meiosis. So, some similarities. They both produce new cells. They both start with one parent cell. And they both go through prophase, metaphase, anaphase, and telophase, what we call PMAT. And here are those two pictures. And I'm going to go through meiosis the same way I did for yesterday's with the mitosis. And any similarities are going to be just plain written in black type, and any differences are going to be highlighted in yellow. So on your notes, let's take a look at that quickly. I want you to fill out what happens in each box here, but only the ones that are different. You don't need to write if it's the exact same as what happens in mitosis unless you want that extra information. Okay. So interphase. Interphase is the exact same as meiosis. I have color-coded the chromosomes so you can see which ones come from the mom, which one come from the dad. But G1, same thing. It is going to be when the cell grows, makes more organelles, checks its DNA. S phase, or DNA synthesis, is when the DNA replicates. Look how the chromosomes have turned into X's because they've replicated, made another copy. And then G2, cell grows a little more, checks its DNA again. Okay, here is showing the 46 human chromosomes again. Before, they were written in random colors to show the difference between chromosome 1, 2, 3, 4, 5, etc. And now they are drawn to show that blue represents chromosomes from the father, red represents chromosomes from the mother. And each pair shows homologous chromosomes. So homologous means that pair where these are both chromosome 1, but this one is from the father, this one is from the mother. Note that these have the same genes, but different variations. We're going to talk about that in Unit 4, but as an example, let's say that on this chromosome there's eye color, and your dad has a gene for brown eyes, and your mom has a gene for blue eyes, okay? So you will have genes for both of those traits, but only one of them is going to show up, and you'll see why in Unit 4. Okay, here is when they have replicated. So back and forth, chromosomes have turned into X's because they have doubled. Each one has made a copy of itself. So we now have 46 replicated human chromosomes. And for the meiosis drawing, I'm going to choose randomly chromosomes 1 and 3 to fill out our cells. Okay, so here's meiosis 1. Remember that any similarities between mitosis and meiosis will just be written like that, and any differences will be highlighted in yellow. Okay, here's prophase 1. Just like mitosis, DNA condenses into tightly coiled chromosomes, and the nuclear envelope breaks down. Centrioles move to opposite poles, and spindle fibers form. The new thing is that homologous chromosomes, look at them, see how they're actually kind of mixed up in their colors? That is because they have done what is called crossing over. Crossing over is when homologous chromosomes exchange genes and mix up their DNA. And this is for maximum variation. So here we have chromosome from your father, chromosome from your mother. They mix up. This is going to happen way more than what I have drawn, where it'll be lots of bands of red, lots of bands of blue, but just to show there were 4 exchanges, and that is crossing over. You can see here, here's also where it literally crosses. This is the 2 chromosomes forming a tetrad when they touch, and then they exchange DNA. Okay, metaphase 1. So homologous chromosomes form tetrads. Notice that strange looking thing where the chromosomes are actually in a group together of 4 chromatids. And then just like in mitosis, the spindle fibers attach to each chromosome tetrad, and then the chromosomes line up along the cell center or the equator. Okay, anaphase 1. So the chromosomes separate to opposite sides or poles of the cell. The thing that is different about this is that the sister chromatids are still together. So the tetrad has separated, but the chromatids are still together. Okay, telophase 1, nuclear membranes start to form. Chromosomes begin to uncoil, spindle fibers fall apart. This is just like telophase and mitosis, except for there's the mixed up crossing over chromosomes. Cytokinesis 1, this is when the cell formally, officially divides into two because the cytoplasm divides between two daughter cells, each with a genetically unique nucleus. So this is unique about meiosis, is that these two cells are not identical. They have different DNA. And then the cells enter interphase, begin the cycle again. Okay, so now we are in meiosis 2, the second part of meiosis. So prophase 2, just like prophase 1 and prophase in mitosis. Metaphase 2, just like metaphase in mitosis, spindle fibers attach, chromosomes line up. Anaphase 2, just like mitosis. Anaphase, where the sister chromatids separate. Okay, notice how now the sister chromatids are separating. Here's telophase 2, nuclear membranes start to form, chromosomes uncoil, spindle fibers fall apart. And then cytokinesis 2. So this is when the cytoplasm divides and the nucleuses are genetically unique. So 4 cells have been created and none of them are genetically identical. They are all unique. And the cells do not enter interphase and begin the cycle again because they have created what they needed to, the sperm or the egg, depending on male or female. Here's an interesting thing about meiosis in the sperm versus egg. So here is the spermatogonium, which is the cell that's going to become 4 sperm. And notice how in meiosis 1, it divides into 2 cells, and then in meiosis 2, it divides into 4 cells, which are then the sperm. Cool? Here is the oogonium. Oo means egg. There's primary oocyte. So in meiosis 1, this oogonium is going to turn into 2 cells, right? But this first polar body actually doesn't ever become an egg. Most of the cytoplasm goes to this secondary oocyte. And then meiosis 2 only occurs for that secondary oocyte, which then does meiosis 2. And then there's a second polar body. Both of these things are going to get just reabsorbed by the body, not used. They won't ever become fertilized. And then here's the ootid. So this is the ovum. This is what becomes the egg. So notice that 1 spermatogonium becomes 4 sperm and 1 oogonium becomes 1 egg. So 4 versus 1. And that's because the egg is the one that has all of the cytoplasm, all of the organelles. The sperm is pretty much nothing except for a packet of DNA and then this tail so that it can propel itself to the egg. But the egg has a lot in it that is going to prepare the zygote. Okay, so here is showing some similarities and differences because what you are going to do for your assignment is identify the similarities and differences. So you have to do at least 3 mitosis only and meiosis only, and then at least 2 for both, what both have in common. There's actually way more than 3 and 2 for both of those. And here are the questions that I want you to consider in order to figure out what's similar and different. So, how many cells does each process start with? Little hint, look, they both start with 1. How many cells does it end with? Okay, here's another hint. Look at how many mitosis ends with. Look at how many meiosis ends with. Are the daughter cells genetically identical to each other? So look at mitosis. Look at those daughter cells. Are they looking identical or unique from the parent cell? And are they looking identical or unique to each other? And then, yep, same thing for meiosis. Are these unique? Are these genetically unique or are they genetically identical both to each other and to the parent cell? What type of cells are created? So that means, are they haploid or diploid? And that also means, are they somatic cells or reproductive cells? And then where does this process happen? What type of cells? So when you're done with that, I'm going to give you a little hint. Here's the key. and you can either pause the video and go back and check on that, or you can go to the link to this PowerPoint. So good luck on finding the similarities and differences. I really, really recommend, I want you to watch this video. It is comparing side by side the difference between mitosis and meiosis. Here's the YouTube video, and then I put it on Edpuzzle, which is basically, if YouTube is blocked, you'll be able to see it. And that shows side by side mitosis versus meiosis, and it is very helpful.